Hello, everyone. Welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It is our great pleasure today to host Dr. Chieh-En Tseng. Dr. Chieh-En Tseng is an instructor in radiology with expertise in magnetic resonance imaging and positron emission tomography. Her research focused on using neuroimaging to understand the biological underpinnings of psychiatric symptoms. Through advanced imaging approaches, she aims to identify neurobiological biomarkers that can inform interventions for psychiatric disorders. Ultimately, her work seeks to reduce symptom burden and improve the quality of life for individuals affected by these conditions. I would like to remind our audience to address their question at the end of the talk, and without further ado, the stage is yours.
Thank you, Chieh-En, for the kind introduction, and I'm really glad to be here today to share with you our work on using positron emission tomography and magnetic resonance imaging, or PET-MRI, to assess neuroepigenetics and how they might relate to psychiatric disorders and symptoms. Okay, so I'd like to first start with sharing this message that was left by a former service member. I'll give you a moment to read through it. So as you can see, this individual suffered from a range of symptoms following his service, including psychiatric symptoms, and he struggled to get help. This is just one example of how psychiatric symptoms can be burdening to a person. And this, in part, is due to there still being a poor neurobiological understanding of psychiatric symptoms, which makes giving appropriate treatment to psychiatric symptoms still a challenge. We've learned that genetics plays a big role in psychiatric disorders, and there has been specific risk genes that have been identified for specific psychiatric disorders, but it still doesn't paint the full picture. The environment also plays a role, and the interactions between genes and environment may influence the vulnerability of one's risks of developing psychiatric symptoms or disorders. So one way we can assess how the environment impacts gene expression is through epigenetics. So the study of epigenetics is showing how the environment, such as through toxins or stress or your diet or exercise, may change your DNA structure without changing the underlying DNA sequence through epigenetic mechanisms, which in turn regulates whether your genes are turned on or off. We are interested in epigenetic enzymes called histone deacetylases, or HDACs. HDACs are called erasers of epigenetic marks because what it does is it removes these acetyl groups shown in blue circles from histone tails, which causes the chromatin structure to become more condensed, and this leads to more repressed gene transcription. So when there are more HDACs, there are less histone acetylation, and there's less gene transcription. Preclinical work has shown that HDACs are associated with learning and memory, as well as mood, which makes it relevant for psychiatric disorders. With the development of the radiotracer Martinos and using it with PET-MRI, we are able to assess HDACs in the living human brain. So here on the left, I'm showing what Martinos looks like. It's an HDAC inhibitor that is tagged with carbon 11, and that we can use as a radio tracer. It binds to class 1 HDACs, more specifically HDACs 1, 2, and 3. And our work has shown that the HDACs that Martinos binds to regulates genes related to synaptic plasticity, such as BDNF and synaptophysin. So here on the right, I'm showing you a group mean map of Martinosat uptake in healthy adults. So you can see the distribution of HDACs in the living human brain, where it's higher in the cortex, lower in the white matter, and higher in subcortical regions such as the thalamus, caudate, and lower in limbic regions like the amygdala and hippocampus. To begin to understand whether HDACs vary with biological factors of age and sex, we scanned 41 healthy adults ranging from the ages of 18 to 79 years old. We were interested in age and sex given their association with disease. For example, with increasing age, there comes cognitive decline and a higher risk for neurodegenerative disorders. And in terms of sex, there are sex differences in the rates of specific psychiatric disorders. For example, women are more likely to have depression. So first, for age, this is work led by Dr. Nicole Zerker, who I will try to show if we can. Anyways, here. We found that HDACs increased with age in the periventricular white matter. So here I'm showing a statistical map of when we looked across the whole brain to assess if there were any associations between age and HDACs, and this periventricular white matter emerged with a positive association. This finding was confirmed with postmortem work, where we found elevated HDACs 1 and 2 in older adults compared to younger adults, which you can see here in the periventricular white matter. And we didn't see differences in HDACs 1 and 2 between the older and younger adults in the corpus callosum, which we selected as a control white matter region. Given that our findings were in the white matter and we also acquired diffusion MRI, we could use the diffusion MRI data to assess white matter microstructure. And we assessed it with this measure called the generalized fractional anisotropy. And just to give a brief introduction, this value is close to 0 when there is unrestricted diffusion, such as in the CSF where water molecules are not restricted and going isotropically. But when the water is restricted by white matter, then it becomes anisotropic diffusion, and GFA would be closer to 1. So when the white matter is altered, what we see is a reduction in GFA. We found that HDACs in the periventricular white matter was associated with white matter microstructure assessed by GFA, where higher HDACs were associated with lower GFA or less white matter integrity. This was found in the post-hoc region, where we found that age was associated with HDACs and restricted to the white matter. So then we also assessed whether white matter microstructure integrity was associated with age, and we conducted this in a whole brain voxelized manner, and we found that white matter structural integrity decreased with age in this light blue area, which shows a great spatial overlap with the region where we found that HDACs were associated with age. So these findings suggest that age-related changes in HDACs and white matter may be linked. Next, for our investigations in terms of HDAC differences related to sex, we found that there are sex differences in HDAC density. HDACs are expressed more in the amygdala, the frontal medial cortex, the thalamus, and the hippocampus in women compared to men, while HDACs are expressed higher in the cerebellar white matter in men compared to women. Interestingly, some of these regions, such as the amygdala and the hippocampus, are regions that are known to have high gonadal hormone receptors. And given that we know there are sex differences in the rates of psychiatric disorders, this finding of sex differences in HDAC suggests their HDACs might contribute to the differences in vulnerability between men and women to develop psychiatric or neurological disorders. With this work, we also determined that the semi-quantitative measure standardized uptake value ratio, or SUVR, is an appropriate surrogate measure for the quantitative volume of distribution measure, which is the gold standard for assessing radiotracer uptake, but it requires arterial blood sampling. So you can see here how SUVR measures across multiple brain regions in a number of individuals corresponded well with the VT measures. This was across an age range of 22 to 53, and in both males and females. So this was good news to us because in some populations, patients may not be able to tolerate arterial blood sampling. So this means that we are able to use SUVR measures to estimate PET uptake, which doesn't require arterial blood sampling. So our first assessment of HDACs in a psychiatric disorder with Martinos PET-MRI was in schizophrenia. We focus on the dorsolateral prefrontal cortex, given how it's often implicated in schizophrenia. And we see that there is lower HDACs in the dorsolateral prefrontal cortex in schizophrenia. So the middle is the group mean Martinos uptake map of controls. The right is the group mean Martinos uptake map in schizophrenia. And you can see visually how the uptake is lower in schizophrenia in this dorsolateral prefrontal cortex region compared to controls. This finding aligned with findings from postmortem work where we found lower HDACs in the dorsolateral prefrontal cortex in schizophrenia compared to controls. So following this work, we've continued to conduct Martinos PET-MRI studies in different conditions, and I will go into more detail for some of them today. The first one that I will talk about is bipolar disorder. Bipolar disorder is characterized by having extreme mood swings, going from emotional highs called mania to lows called depression. There are different types of bipolar disorder. People with bipolar disorder type 1 have at least one manic episode, which usually occurs before or after a depressive episode. People with bipolar disorder type 2 have hypomanic episodes and depressive episodes. People with cyclothymia have maybe a milder form, and they experience hypomania and mild depressive episodes. Research has shown that HDACs may be implicated in bipolar disorder. So preclinical work shows that HDACs are involved in mania and depressive-like symptoms. HDAC inhibition reduces hyperactivity in models of mania, and it also reduces depressive-like symptoms in preclinical models of depression. Currently used pharmacological drugs for bipolar disorder also act on HDACs. Valproic acid and lamotrigine both inhibit HDACs, and they are often used as mood stabilizers. And lithium, which is also a commonly used mood stabilizer, downregulates HDACs. So given this, we wondered, are HDACs altered in bipolar disorder? And specifically, is it altered in the amygdala, which is a key region for emotion processing? Also, given the involvement of HDACs in cognition, we wondered whether HDACs were associated with emotion regulation in bipolar disorder. For this project, we recruited participants with bipolar disorder and age and sex matched controls. The participants with bipolar disorder met the DSM-4 criteria for bipolar disorder type 1 or type 2 and were between the ages of 18 to 65. The healthy controls were healthy individuals between the age 18 and 65 and were using no psychotropic medications. For both groups, none of them could be using any illicit drugs or marijuana, and they couldn't have any major physical illness, head trauma, or any PET-MRI contraindications. All of our participants completed a Martinos PET-MRI scan on the 3T Siemens TIM trio with a brain PET insert. They were all injected with 5 millicuries of Martinostat. With the scan, we acquired a T1 weighted structural image and the PET list mode data, where we focused on the 60 to 90-minute post-radio tracer injection period. With the T1 weighted structural image, we were able to generate an MRI-based mu map that we used to conduct attenuation correction for the PET data. And we were able to generate the standardized uptake value maps, which we then normalized by the whole brain mean to obtain the standardized uptake value ratio maps, or SUVR maps. Given that our a priori region of interest, the amygdala, is a pretty small structure, we opted to use the geometric transfer matrix method to conduct partial volume correction, which we did using FreeSurfer. So for this project, we scanned 11 participants with bipolar disorder and 11 controls. They were matched in terms of age and sex. And to give a brief description of the medications that the participants with bipolar disorder were on, 6 participants with bipolar disorder were taking lithium or lamotrigine, and 8 were using antipsychotics. And we will try to account for the medication in our analysis. So with the region of interest analysis, we found lower HDACs in the amygdala in bipolar disorder, specifically in the right amygdala, but there's a trend level group difference in the left amygdala as well. We considered the potential of medication being a confounding factor, but we didn't find any association between the uptake of Martinostat in the right amygdala and the chlorpromazine equivalent dose in bipolar disorder patients. We also didn't find any difference between Martinostat uptake in the right amygdala between those who were taking lamotrigine or lithium and those who weren't, or between bipolar disorder type 1 versus type 2. We further conducted exploratory analysis across the whole brain, and we found additional regions that showed lower HDACs in bipolar disorder, including the orbital frontal cortex, the thalamus, and the hippocampus. So this is showing that HDACs are lower in frontal limbic regions in bipolar disorder. The frontal limbic network is involved in emotion regulation, and previous functional MRI studies have shown that this network is altered in bipolar disorder. So our findings suggest that HDACs may contribute to alterations in the frontal limbic network in bipolar disorder. We also assessed whether HDACs are associated with emotion regulation in bipolar disorder and found that higher HDACs were associated with better emotion regulation skills in the prefrontal white matter and the perisylvian region, while higher HDACs were associated with poorer emotion regulation skills in the middle frontal gyrus. So we see associations going in both directions, and it's a bit of a more complex picture, but the brain regions where we find the associations are somewhat related to emotion regulation. For example, frontal regions may be associated in decision making, and superior temporal gyrus is associated with social processing. So to give a brief conclusion about what we've learned in bipolar disorder, we found that HDACs are lower in the amygdala and frontal limbic regions in bipolar disorder. These are regions that are often implicated in bipolar disorder. And we found that regional HDACs are associated with emotion regulation in bipolar disorder. So moving on, I will go to the next condition that we have investigated. We recently investigated neuroepigenetics in autism spectrum disorder. Autism spectrum disorder is characterized by having problems with social communication and social interaction, as well as having restrictive, repetitive patterns of behavior, interests, or activity. These symptoms often begin, not often, these symptoms begin in early childhood. The prevalence of ASD is increasing as shown in this top graph here. And in 2022, the statistics show that 1 in 31 children were diagnosed with ASD. ASD is also 3.4 times more often diagnosed in boys compared to girls. Research has shown that HDACs are implicated in ASD. In humans, maternal use of valproic acid, which we previously mentioned, inhibits HDACs during pregnancy, leads to an increased risk of ASD in the child. Genetic work has shown several risk genes for ASD encode proteins that interact with HDACs. And postmortem work shows that there's altered acetylation near HDAC genes in both idiopathic and syndromic ASD. In terms of preclinical work, we found that prenatal HDAC inhibition leads to ASD-like symptoms in the offspring, and perhaps a bit counterintuitively, postnatal HDAC inhibition in models of ASD actually rescues social deficits. So collectively, these findings show that HDACs are maybe altered in ASD, but whether this is the case in the living human brain is still unknown. Our own data in healthy adults show that HDACs in the anterior cingulate cortex are associated with emotional skills. This is measured by the perspective taking subscore of the interpersonal reactivity index. So higher HDACs in the anterior cingulate cortex is associated with better perspective taking skills. We therefore hypothesized that HDACs in the ACC might be altered in ASD. So for this study, we aim to investigate whether HDACs are altered in ASD, and we wondered whether there are sex-specific changes of HDACs in ASD, given that it is diagnosed more often in boys. And we wondered whether HDACs are associated with language and ASD symptoms. We selected the anterior cingulate cortex as the a priori region of interest, given the findings from our preliminary data. So for this work, we recruited participants with ASD who met the DSM-5 diagnosis of ASD, which was also confirmed with 2 other diagnostic tools. There were between the ages 18 to 40 years old, and we included participants across the spectrum, so they had a range of IQ, and we included anyone who had an IQ of above 35. But we excluded for epilepsy, given that it might impact HDACs. The control group consisted of healthy individuals between the ages of 18 to 40 who weren't using any psychotropic medications. Both groups could not be using any HDAC inhibitor, any illicit drugs or marijuana, and they couldn't have any major physical illness, head trauma, or PET-MRI contraindications. For this project, we had all our participants complete a training visit where they are able to lie inside the actual PET-MRI scanner that we use for the scanning and listen to the noises that the scanner makes. So this will allow them to become accustomed to the scanner environment and make sure that they are comfortable with completing a scan in that scanner for around 40 to 50 minutes. We found that this increases the comfort level for participants, and it also allowed us to acquire high quality imaging data. So similarly to the bipolar study, we scanned all of our participants with the 3T Siemens TIM trio with a brain PET insert, and they were injected with 5 millicuries of Martinostat. We generated the SUV map as described earlier. And for the region of interest, we took the anterior cingulate cortex parcellation from FreeSurfer. To characterize their social difficulties and restrictive and repetitive behaviors, we asked their caretakers to complete a couple of questionnaires. So we assessed social difficulties with the social withdrawal subscore from the aberrant behavior checklist, and the social awareness, social cognition, social communication, and social motivation scores from the social responsiveness scale, addition to. Restrictive and repetitive behaviors were assessed based on the stereotypy subscale of the aberrant behavior checklist, the restrictive interest and repetitive behavior score from the social responsiveness scale, second edition, and the total score of the repetitive behavior scale revised. Additionally, we assessed language in our participants with ASD with the Peabody Picture Vocabulary Test 4th edition. This test assesses receptive vocabulary, where the examiner will ask the examinee to point to a specific item, which is shown in the figures that they are displayed. So for example, this examiner may be telling the examinee to point to the drums, and then the examinee is pointing to the drums. For this project, we scanned 21 participants with ASD and 23 controls who were matched in terms of age and sex. For the region of interest analysis, we found lower HDACs in autistic males in the anterior cingulate cortex. This was found by a group by sex interaction, and the significant interaction was driven by a significant group difference between ASD males and control males in martinostat uptake in the ACC, while there was no group difference between ASD females and control females in this region. We also conducted exploratory whole brain analysis to see if there were group differences in other regions, and we found that HDACs were lower in the insula, hippocampus, superior temporal gyrus and sulcus, and orbital frontal cortex in ASD compared to controls. These regions are involved in social emotional processing, which is impacted in ASD. In terms of the assessment of associations between HDACs and language and ASD symptoms, we only found an association between HDACs in the ACC and receptive vocabulary in ASD participants. So lower HDACs were associated with weaker receptive vocabulary in ASD. We considered whether the findings were driven by the individuals with IQ lower than 85, but we were able to obtain similar findings for both the group comparisons and the associations when we conducted the analysis, including only individuals with IQs above 85. So in conclusion, we found that HDACs are lower in ASD, specifically in regions that are involved in social emotional processing, and that regional HDACs are associated with language skills in ASD. So to give a brief summary of what we learned so far, we kind of see that HDACs are altered in a disorder-specific manner. We found that HDACs are lower in the dorsolateral prefrontal cortex in schizophrenia. The dorsolateral prefrontal cortex is a key region for executive function and is often implicated in schizophrenia. In bipolar disorder, we found that HDACs are lower in the amygdala and frontal limbic regions, which are also often altered in bipolar disorder and related to emotion regulation. In ASD, we found that HDACs are lower in the hippocampus, insula, and superior temporal gyrus, which are regions that are engaged in social emotional processing and is impacted in ASD. So while we don't know what genes are being impacted by the alterations in HDACs in these disorders, we find it interesting that HDACs are altered in regions that are relevant to that specific disorder. So for the next part, I'd like to share some of our ongoing work. So we are now assessing neuroepigenetics in combat veterans with repeated blast exposure. Service members are exposed to an environment of repeated low-level blasts during training and in combat. These low-level blasts can come from breaching explosives, as shown in this video here, as well as from handheld or shoulder-fired arms. You may have noticed that these blasts are often from their own weapons and not from the enemy's weapons. Low-level blasts cause overpressure waves that are below 4 pounds per square inch, or PSI. To put this in perspective, a blast wave of 1 PSI can shatter a window. While single low-level blast exposures don't cause immediate acute injury, the impact of repeated low-level blasts on one's brain health is still not well understood. If you remember the message that I showed you in the beginning of this talk, that individual endured high levels of repeated blasts during his career. It's being now increasingly recognized that service members may not be appropriately protected from repeated blast exposure. It's thought that blast waves can enter the brain through openings in the head, such as the orbit or ear canals. Blast waves can come directly from the blast, as shown by this incident wave, or it can come by being reflected off of walls or the floors and come as a reflected wave. So sometimes it's not necessarily the person that is closest to the blast that has the most exposure. Let me show you a simulation of what happens in the brain when it is exposed to blast. You can see the amount of shearing that is estimated to occur in the brain when it is exposed to blast, and imagine how repeated exposures to blast may cause brain injury. Service members or veterans who have experienced blast exposure during training or in combat have reported a range of symptoms, including memory problems, post-traumatic stress, anxiety, and depression. A few years ago, we conducted the ReBlast pilot study, which was led by Dr. Brian Edlow, and one of the goals was to identify neuroimaging correlates of repeated blast exposure. We scanned 30 United States Special Operation Forces personnel with structural and functional neuroimaging and found that structural and functional neuroimaging measures of the rostral anterior cingulate cortex was associated with the levels of blast that they experienced. This is interesting because the rostral ACC sits above the orbits, which is where it's thought that blasts would enter to the brain. Preclinical work shows that blast exposure alters epigenetics. In the hippocampus, following repeated blasts, HDAC increases and histone acetylation decreases. So if you'll remember, the more HDACs there are, the less histone acetylation there is. The hippocampus is known for its key role in memory, and this is a domain where the service members who were experienced repeated blast exposure had problems with. Histone acetylation is also decreased in the rostral anterior cingulate cortex following blast exposure in preclinical models. And this is the region that we found to be vulnerable to repeated blast exposure. So given this, we are currently conducting a study to assess if HDACs are associated with levels of repeated blast exposure, memory difficulties, and symptoms of PTSD, anxiety, and depression. The a priori regions of interest that we will focus on are the hippocampus and the rostral ACC. We are recruiting combat veterans with high repeated blast and low repeated blast. Blast exposure will be determined based on the generalized blast exposure value, which is a self-report questionnaire. All participants will complete a Martinos PET-MRI scan to assess HDACs, a neuropsychological battery to assess memory, self-report questionnaires to assess symptoms of post-traumatic stress, anxiety, and depression, and we will obtain blood samples to assess blood biomarkers such as BDNF. We think that Martinos PET-MRI has the potential to identify brain injury caused by repeated blast exposure. This is definitely a huge team effort. This is a collaboration between the Martinos Center, which includes the neuroimaging and clinical team, as well as collaborators at home base, where we are lending their neuropsychological expertise. We have consultants from Mount Sinai and the Vanderbilt University to give us their expert guidance on TBI research. And we are also working with a community advisory board that is made up of veteran clinicians and scientists and real life experience consultants who will advise us on like what measures we should be focusing on, like what's most important to them. And we're hoping that this work will be able to contribute to the development of either diagnostic tools to identify brain injury from repeated blast, or maybe even down the line treatment methods. Yeah. So with that, I'd like to acknowledge all my mentors and collaborators and the funding sources and all our participants and their families for making this work possible. And thank you for your time.